This is simple squamous epithelium. So far in previous videos, we talked about the characteristics of epithelial tissue. We talked about the functions of epithelial tissue. We talked about how we name them based on the number of layers plus their shapes. And now that finally leads into we can study the individual types. So we're starting with simple squamous epithelium. Remember, simple meaning one layer of cells. Squamous meaning thin and flat. So you use the three terms to describe the tissue. Where do we see simple squamous epithelium? Remember, simple is to move things. Squamous is thin, flat cells. Good example is alveoli of the lungs. So I exchange those gases. When we look at histology or the slides of the microscope for simple squamous epithelium, I use this little picture to go back to show the arrow. We are looking at it like you're looking at the tiles of the floor. So other microscope slides, when we look at epithelial tissue, would look more like a side view. So when we look at the microscope, it looks like it's multiple layers, but not when you look down on the floor, just looking at the top of the tile. So you're looking at essentially the apical surface. Three different views depending on what microscope slides you have. It's just like anything else. I look different than you, but I still have a nose. It just looks different. So these three slides are the same. It's just all simple squamous epithelium. So we're looking at the top of it. All of these are individual cells. So I have, 100 slides or 100 cells in this picture here on the top left. Remember, the dark dot in the middle indicates the nuclei, and then the surrounding line around it is my cell membrane, plasma membrane, or plasma lemma. Any of those three terms work. And then we're just looking at the material inside the cell, which then is the cytoplasm. If I go down to this one, cells are a little spread out in this microscope, but remember we have cellularity, tightly packed cells. So all the individual cells, and I can see them distinctly because I have that dark dot in the middle, the nucleus, the cytoplasm within it, and then the boundary, my cell membrane. I like this one the best. I think it's the most consistent slide you'll see across text, but this is all showing that cellularity. And here's some nice cells here as I wrap around the cell membrane, the middle dark dot, the nucleus, and then cytoplasm is the material in it. If we had a more powerful microscope like an electron microscope, we could see some of the organelles potentially. But for a light microscope, it's going to be pretty universal that we're just going to be able to see the nucleus and the cell membrane so we can see from cell to cell. Simple squamous epithelium.